Protocol: Phenol-Chloroform extraction of prokaryotic DNA. Deoxyribonucleic acid, DNA, extraction is the process by which DNA is separated from proteins, membranes, and other cellular material contained in the cell from which it is recovered. The simplest cells, such as bacterial cells, are prokaryotes. These prokaryotes comprise a lipid bilayer outer membrane and a cytoplasm containing a circular chromosome, proteins, inorganic salts and metal ions, sugar molecules, and other elements of cell machinery. A large number of different protocols for the efficient isolation of highly purified DNA from both eukaryotic and prokaryotic cells is extant. Many of the available DNA extraction procedures have common elements. Indeed, the extraction of DNA generally follows three basic steps. Lyse: break open the cells. Separate the DNA from the other cell components. Isolate the DNA. Phenol-Chloroform extraction of DNA. A phenol-chloroform extraction is a liquid-liquid extraction. A liquid-liquid extraction is a method that separates mixtures of molecules based on the differential solubilities of the individual molecules in two different immiscible liquids. Liquid-liquid extractions are widely used to isolate RNA, DNA, or proteins. The extraction of nucleic acids involves adding an equal volume of phenol-chloroform to an aqueous solution of lysed cells or homogenized tissue, mixing the two phases, and allowing the phases to separate by centrifugation. Centrifugation of the mixture yields two phases, the lower organic phase and the upper aqueous phase. Principle: the organism to be used should be grown in a favorable medium at an optimal temperature, and should be harvested in late log to early stationary phase for maximum yield. The genomic DNA isolation needs to separate total DNA from RNA, protein, lipid, etc. Initially, the cell membranes must be disrupted in order to release the DNA in the extraction buffer. SDS, sodium dodecyl sulfate, is used to disrupt the cell membrane. Once cell is disrupted, the endogenous nucleases tend to cause extensive hydrolysis. DNA can be protected from endogenous nucleases by chelating Mg2++ ions using EDTA. Mg2++ ion is considered as a necessary cofactor for action of most of the nucleases. Nucleoprotein interactions are disrupted with SDS, phenol, or proteinase K. Proteinase enzyme is used to degrade the proteins in the disrupted cell soup. Phenol and chloroform are used to denature and separate proteins from DNA. Chloroform is also a protein denaturant, which stabilizes the rather unstable boundary between an aqueous phase and pure phenol layer. The denatured proteins form a layer at the interface between the aqueous and the organic phases, which are removed by centrifugation. DNA released from disrupted cells is precipitated by cold absolute ethanol or isopropanol. Materials and reagents: tris base, proteinase K, phenol-chloroform 1:1, 200 proof ethanol, aranose, ethanol, SDS, EDTA, tryptone, yeast extract, NaCl, Lb medium, Te buffer, lysis buffer. Equipment: tabletop centrifuge, 1.5 ml Eppendorf tube. Incubator. Procedure: transfer 1.5 ml of the overnight E. coli culture, grown in Lb medium, to a 1.5 ml Eppendorf tube and centrifuge at max speed for 1 minute to pellet the cells. Discard the supernatant without disturbing the cell pellet. Resuspend the cell pellet in 600 ml lysis buffer and vortex to completely resuspend cell pellet. Incubate 1H at 37 degrees Celsius. Add an equal volume of phenol/chloroform and mix well by inverting the tube until the phases are completely mixed. Spin at max speed for 5 minutes at RT. All spins are performed at RT, unless indicated otherwise. There is a white layer, protein layer, in the aqueous, phenol-chloroform interface. Carefully transfer the upper aqueous phase to a new tube by using 1 ml pipette man. To avoid sucking the interface, use 1 ml tip with wider mouth. Cut 1 ml tip mouth about 2 mm shorter. Steps 4 to 6 can be repeated until the white protein layer disappears. To remove phenol, add an equal volume of chloroform to the aqueous layer. Again, mix well by inverting the tube. Spin at max speed for 5 minutes. Remove aqueous layer to new tube. To precipitate the DNA, add 2.5 or 3 volume of cold 200 proof ethanol, store ethanol at -20°C freezer, and mix gently. DNA precipitation can be visible. Note, DNA precipitation may simply diffuse, which is normal. Keep the tube at -20° for at least 30 minutes, the longer the better, and then spin it down. See steps 15 to 16. You should see DNA pellet. It looks transparency when it is wet and turns to white when it becomes dry. Incubate the tube at -20°C for 30 minutes or more. Spin at max speed for 15 minutes at 4°C. Discard the supernatant and rinse the DNA pellet with 1 ml 70% ethanol, stored at RT. Spin at max speed for 2 minutes. Carefully discard the supernatant and air dry the DNA pellet. Tilt the tube a little bit on paper towel. To be faster, dry the tube at 37°C incubator. Resuspend DNA in TE buffer. Note, large amounts of RNA will be present in the DNA sample. So, for subsequent reactions, for example, to digest plasmid DNA, add 1 to 5 μl, 1 mg ml1, RNase to the digestion solution to completely remove RNA. Or, add RNase directly to lysis buffer with a final concentration of 1 mg ml1. Isolated genomic DNA can be subjected to agarose gel electrophoresis. Expected results: white strands of DNA precipitate. On gel, bands with smear patterns from high to low molecular weight range can be seen. Most of DNA fragments accumulated at high molecular weight, not degraded. Most of DNA fragments small, DNA might have got degraded.